Our next speaker is Dr. Ronald Kleinman. He is a physician-in-chief at Mass General Hospital for Children. He's chair of the pediatrics and chief of the pediatric gastroenterology and nutrition unit at MGH, and the Charles Wilder Professor of Pediatrics at Harvard Medical School. His major areas of research interests include gastrointestinal immunology, nutrition, support of infants and children, and nutrition and public health policy. Dr. Kleinman also serves as in a number of medical societies, many in leadership roles, and on the boards of the David Ortiz Children's Fund, the Global Children Nutrition Foundation, and Project Bread. Dr. Kleinman is the author of multiple editions of the Academy's Pediatric Nutrition Handbook and editor-in-chief of the current Pediatrics Reports. Dr. Kleinman is a graduate of Trinity College in Hartford, Connecticut, and earned his MD from New York Medical College and completed his residency and chief residency in pediatrics at Albert Einstein College of Medicine in New York, and his fellowship in pediatric gastroenterology and nutrition at MGH, Harvard Medical School. Please welcome Dr. Kleinman.
Well, you can see from that introduction that I need to start this off with a disclaimer. You know, in broadcast journalism, the British make a distinction between reporters and readers. Reporters are individuals who go out and do the investigation and then report their results. Readers are individuals who read what the reporters have written. Ladies and gentlemen, I'm a reader. Chris McDougall, the director of the LURIE Autism Center, has asked that I stand in for him today. Happily, he's in California right now receiving a Lifetime Achievement Award from the American Academy of Child and Adolescent Psychiatry, and so that's why I'm here and he's not here. I would also like very much to thank Paul Marcus and the Autism Consortium for inviting us to speak today, and certainly the Nancy Lurie Marks Family Foundation, Nancy Lurie Marks, and I see Shutt sitting here, as well as I think I saw Dick Denning in the back. Both of them have been absolutely wonderful, and you'll see their influence as I go through this presentation. So the goal of the LURIE Center is to engage the many thousands of children and adults that have come through what was the Ladders program, which is now part of the LURIE Center over these many years, together with the scientists that are in the center now and those scientists that we collaborate with locally, nationally, and internationally, to better understand the mechanisms and the causes that lead to the spectrum of autism disorders, and of course ultimately to find a cure or cures. This is a list of the current collaborators with the LURIE Center, and you can see that they cross university boundaries, hospital boundaries, as well as multiple disciplinary boundaries and departments in the various institutions. There are really, at this point, 4 areas of focus at the LURIE Center. And I'm going to spend the next few minutes, and I think I probably will finish a bit early, so you'll be able to catch up. These 4 areas, I'm going to spend just a few minutes going through each of these 4 areas for you to begin to describe some of the work that the LURIE Center is doing. We use the word program, project, grant because that's what Shutt told us to call these. These are focus areas that have been developed with a goal towards developing data and evidence that can then lead to further grant support. And so this does seem to be a very apt way to describe these. These are the principal investigators in the first of these focus areas, the Bioinformatics Database, and they consist really of 2 committees or groups, the phenotyping and the genotyping groups. Julia O'Rourke, who led the development of the database for the Autism Consortium, joined the LURIE Center and is now leading the development of this bioinformatics database. And this is the framework that she's been working from. It's a framework that begins with the patient and allows the patient to provide information about either the child or an adult before they even reach the LURIE Center, and then to add to that information phenotypic information and genotypic information together with neuroimaging. There is a concerted effort by Dennis Wall and others to streamline the process of developing the phenotypic information, which is generally developed using various instruments like ADOS and ADIR, streamlining and simplifying those instruments so that rather than spending 2 to 3 hours of filling these out, they can be done in relatively few minutes, streamlining the ADOS down to 2 questions and the ADIR down to 7 questions. The genotypic data will come from blood samples and skin biopsy samples that will be obtained on all patients who consent and come through the center. And then I already mentioned the neuroimaging, and we'll come back to that. The second project is in communication, and that, of course, is based on the 30-year experience in working with children and adults at the LURIE Center, the Ladders program, to help develop better communication skills with some success, but as Lenny mentioned, limited success. And here there's another group of principal investigators, and as with the previous project, these are individuals who come from many different institutions across town. We are going to focus on autistic individuals with minimally verbal skills, those who are moderately impaired. My understanding is that most of the work to date has been done in those who are actually quite high functioning. The work here is going to take those who are much more impaired. And to use multimodal neuroimaging modalities such as MRI and magnetoencephalography and the Connectome, and I'll mention that in just a minute. And these individuals will undergo intensive speech therapy for a period of 4 months. Twice a week by speech therapists, and then during the off days and the weekend days, parents will be implementing these therapies. And the imaging will be done then after the therapy is completed to try to gain some insight on the effect of these therapies on ability to communicate. One of the great advantages is the collaboration with the Martinos Center, and this imaging, which I understand is unique in the world, allows one to understand the neurobiological basis of the nonverbal brain and then use this knowledge to develop effective interventions. And this is a picture of the imaging machine at the Martinos Center. There are 2 such machines. One is a dummy machine to allow children to become acclimated to the machine before they actually have to go through the real imaging process. And this is such an image, and I have to confess that there are many days in the past few years when this was actually the mental image I had of my own brain. But it turns out that the brain is much more impressively organized than most of us thought. It's not simply a tangle of synaptic connections, but is organized in these bundles, much as you'd imagine the communications of a major city are organized if you were able to peer beneath the pavement. The third project is based on observations from autopsy studies of adults who have died of other causes and who've been autistic. And some of these brains demonstrated acute inflammation. And building on that observation, a group of investigators at the LURIE Center have developed both animal studies as well as human studies to look at this impact of inflammation and its effects when superimposed on a vulnerable genotype. Bill Carlson, for example, at the McLean Hospital, has developed an animal study in which he looks at 3 groups of pregnant mice, one of which has been exposed to lipopolysaccharide intraperitoneally during pregnancy, a second, poly-IC, which is a surrogate for viral infection, and a third, turpentine, or a toxic approach, and then looks at the offspring of these mice through imaging. And a unique imaging tool has been developed at the Martinos Center by Jacob Hooker, and this is a ligand that recognizes activated microglia. And I'll show you a picture of this in just a minute. In addition, there is a human study that will look at autoimmune disorders in families and the potential for that developing into inflammation in the brain of individuals with autism. And in this study, again based on observations that Andy Zimmerman and Chris McDougall have made, that individuals with autism have a higher familial incidence of autoimmune disorders, even though they themselves may have no symptoms of autoimmune disorders. This study will take adults with autism, and these are adults who have first or second degree relatives with a dense history of autoimmune disorders, and compare them to those from families without autoimmune disorders and to controls without autism, and we'll put them through PET scans using this specific microglial ligand. And the goal here is to determine whether or not inflammation is playing a role, and then, if it is, to think about and apply potential neuroimmune modulating treatments. This is an example of how this imaging can be applied. The chemical signature of the ligand is in the lower right-hand corner and in the blue box. If you can see those arrows, particularly the red outline there, those are the ligand bound to activated microglia in 2 conditions with neuroinflammation. And then the last of the 4 areas has actually been described quite well already by other speakers this afternoon, Steve and Mrganka. And this is the process of high-throughput, individualized examination of potential therapies for autism spectrum disorders. Jim Gazella and others from the Center for Human Genetic Research at the MGH are the principals in this project, and as you've already learned, this will utilize skin cells from the biopsies that will be obtained from patients at the LURIE Center. These are reprogrammed to develop into neuronal cells, and these cells will then be plated and serve as and will be interrogated then by small molecules and other approaches, both to get insights into functioning as well as potential therapies on an individualized basis. Transgenic models will also be developed in order to better phenotype and characterize the genetic changes that will be observed, ultimately leading to novel medical treatments. This is an example of plated cells, healthy control, and then neuronal cells from fragile X mutation. And I think the differences are fairly obvious. The highly connected and clustered neurons in the healthy control are much more spread out and many fewer connections in the fragile X mutation. And we'll, I think, learn quite a bit from this approach. So I'm going to conclude. I've gone through this, as I said, very quickly. These are the individuals who are located at the LURIE Center. We're very pleased. Chris has been in place just for a year, and I think that they've made tremendous progress in that period of time, both in developing these very important collaborations as well as a framework for clinical treatment and research. And we look forward to hearing from you and to any comments or questions you might have. There are a number of people here today who are probably from the LURIE Center and from these projects who are better equipped than I am to answer your questions, and so I'm glad to try to do that. I thought it would be really good for me to have at least one original slide in here, and so these are my granddaughters, and I try to end every talk I give actually with a picture of my grandchildren. So thank you very much.